This is sort of what we would collect with that suction sampler. It has been used widely to look at juvenile lobsters. Because juvenile lobsters are really hard to catch by hand. Very, very quick. But they'll also catch the diversity of other species, particularly slow moving ones. And they'll suck up dogs too. Water quality sampling. Anything we do around the cage site, water quality is really key. Actually, anytime we sample diversity. The more we know about water temperature, the depth we're at, how much light there is, the more information we have in terms of what the habitat is. Very simple instrumentation for water, for reporting water quality. This is a mini logger, essentially a little computer chip with some sensors in it. Very, very durable. Throw this in the water for months. Just program it, throw it in the water for months. They'll take the temperature, they'll take what depth it's at. Someone will even tell me how much light there is down there. And I just retrieve it, get back, and then download the computer. It's all kept very simple, just battery powered. If I want to know a little bit more about, well, these are about $250, so you see one of these is shoreline. You can talk to me now. Because I've lost a few already. You want a little bit more information? You do something like this. A little bit bigger. It's a multi-parameter sonde. Sonde is just a French word for probe. I don't know, it's oceanographer, so we like using it. Essentially, this one can do not only temperature and depth, but it can do salinity, pH, chlorophyll, turbidity. It has a whole series of fancy probes in the end that will do that. All these new ones use laser light and actually do chlorophyll counts and turbidity counts and oxygen counts as the way. Very sophisticated piece of information. Again, the same concept. This one has a handheld. I can just throw it in the water and get the measurement right away. Or I can detach this and throw it in the water and leave it there for months, and it'll just record as often as I want. That's about 12,000. I don't want to use these. So, incidentally, this thing that looks exactly the same, or very close to the same, that sort of replaced this. This is a Niskin model. It's designed to take a water sample at any depth. So you can attach a line to it, and essentially drop it down a certain depth. You send a heavy weight down, and the heavy weight goes, and it closes it up. Bring that back up, the water sample there, so okay, this is my water quality. You put the sample in different bottles and you take it all back to the lab and do the analysis. The problem is that you've got to do a lot of, you want to know different depths and different areas. It's way too much work, so it's been pretty much replaced by that type of instrumentation. What else? Okay, let's wrap up. Okay, so no big huge words of wisdom, except just to say that in our backyard, there's lots of biodiversity. There's just as much value and diversity in this area as you can see in tropical forests, in tropical forests and coral reefs. It's just that we haven't really spent a lot of time discovering it. We look underneath the water, even a simple area like just a rock, and St. Croix River is just covered with life. There's at least 20 species in that account. You can see the big forest mussels. I was going to say Melotus, and that's the last item. But anyway, just a simple rock full of life. You just have to take a look. Species will use any habitat they can find. This is a lobster hiding under a rock. This is a man-made habitat. This is a sculpin. So species will occupy and take advantage of any habitats that's there for protection.
Distracted by the beauty of it, it's the colors.
Yeah, it's really quite. It's really quite something when you get down into the connection of what these other things are. The only other thing I want to say is that we really do have to take the effort to look at our own backyards. All it takes is you get down, take a closer look, and maybe sometimes get a little bit dirty. The value of the biodiversity in your own backyard is is just as valuable as what we see locally. And certainly a lot more tangible in terms of tourism and trade and future projects. And with that, I would say thank you. And this is just a picture of not nearly all the people that helped me over the last few years. This is all the technicians and colleagues and just people that just come out and help us. There's no way I can do all this by myself. It really is a team. And are there any questions? And if anybody wants to take a look at this, come down and take a look at some of this stuff, and please feel free if there are any questions.
I've been on the base for about 32 years, starting here 32 years ago, doing some science, and that was sort of just before aquaculture came in. And my non-scientific observations that the water and clarity of the water and that stuff has changed. And I'm wondering if when you look at historic data, if you're seeing species change in biodiversity or changes in biodiversity. Are you able yet to make any comments about changes?
Water quality?
Yeah.
Yeah, that's one of the things we definitely want to look at is try to get back to some of the water quality samples that were there 30, 40 years ago to see how temperatures change, see if there's changes in plankton communities. We have to count leaks on this. I probably haven't spent nearly enough time to say anything about that, but it's certainly something that has to be.
The other question, if I could, is were you able to, or has anyone at the station done some studies up by the port and the gravel and the siltation and that's what's in the water pre-quarry and after, or post-quarry?
Yeah, I know there's some research that was done by Art McKay, what was that, like 20 years ago now? There's some coastal dive studies. And it would be, good opportunity to use that data and go back and review. It's just requires the time and effort just to go up and try and figure out what the real question is.
Can you just mention some of the issues you're trying to speak to?
Yeah. So a lot of the biodiversity work that we're doing now is just to establish baselines because we really don't know. We have species lists, but we really don't know how those species communities can change over time, in a short period of time period, and a long time period. So we really have to go back, see what data is available, try to repeat it as closely as we can so we get a sense as to how things are changing over time and relate that to, like this gentleman was saying, relate that to things like water quality. So a lot of the biodiversity work that needs to be done is just a lot of that baseline, just to get a basic understanding of what is natural variation and what's variation is actually caused by something else that's man-made. So that's just in terms of, and then in terms of the aquaculture site, that's one activity where there's wild species that inhabit the area, and I think there's a huge opportunity to learn from the diversity to see what's happening in interactions between something like industrial aquaculture, where we not only have habitat being introduced there, we also have nutrients being introduced there to see if there's anything we can learn from the wild species, just to better understand what's happening in aquaculture.
What major studies did you have to go through to get into research like this? Like, did you have to get a master's or what specific major did you go through?
Yeah, I have a PhD in biology. Actually, I would say it's just biology, but I did specialize in evolutionary biology, systematics and taxonomy. I worked on flatfish for a period of time. Good education, ecology, marine biology, that kind of biology. People that help me have a variety, work with me and lead different projects in a variety of backgrounds, whether it's just basic ecology or specific to marine biology, specific to aquaculture or animal rearing.
Yes, I wondered if you have any idea of the numbers of alewives presently in the bay compared historically to before they cut off the the ability for them to go up the same road.
Yeah, I don't have that specifically, and the only information I have is with that demersal trawl that I was showing you earlier. I'm not convinced that that demersal trawl catches things like alewives that efficiently. So when I'm looking at comparing historical changes, all I can really say is that if I use the same technique that was used 30 years ago, there's the change. But in terms of real numbers, there needs probably a different type of sample.